1. Hormone therapy. Testosterone replacement therapy (TRT) is the primary treatment, helping to develop male secondary sexual characteristics, increase muscle mass, improve mood and energy levels, and maintain bone density. Karyotyping is a laboratory technique used to analyze the number, size, shape, and structure of chromosomes in an organism's cells. It is a fundamental tool in genetics for detecting chromosomal abnormalities associated with genetic disorders, congenital anomalies, infertility, and certain cancers. By arranging chromosomes in a standardized format based on their size, banding patterns, and centromere position, karyotyping allows geneticists to identify numerical and structural chromosomal variations. The process of karyotyping begins with the collection of cells from sources such as blood, bone marrow, amniotic fluid, or chorionic villus samples. These cells are then cultured in a growth medium to promote cell division, as chromosomes are most visible during the metaphase stage of mitosis. A chemical agent such as colchicine is used to arrest cells in metaphase, ensuring that chromosomes are condensed in line for microscopic examination. The cells are then treated with a hypotonic solution to spread the chromosomes, followed by fixation and staining with dyes like Ginza, which highlights specific banding patterns unique to each chromosome. There are different types of karyotyping techniques based on staining methods. G-banding, the most commonly used method, produces a characteristic pattern of dark and light bands that help distinguish individual chromosomes. Other techniques include Q-banding, which uses quinacrine fluorescence, and R-banding, which enhances the visibility of certain regions by reversing the staining pattern of G-banding. High-resolution karyotyping allows for the detection of subtle chromosomal changes by analyzing chromosomes at an earlier stage of mitosis when they are less condensed. Karyotyping is primarily used to diagnose chromosomal disorders. Numerical abnormalities involve changes in chromosome number, such as trisomy 21, Down syndrome, where an extra copy of chromosome 21 is present. Other examples include Turner syndrome 45, X where one X chromosome is missing in females, and Klinefelter syndrome 47, XXY, where males have an extra X chromosome. Structural abnormalities include deletions, duplications, inversions, and translocations, which can lead to genetic disorders such as Cri-du-Chat syndrome, caused by a deletion on chromosome 5, and chronic myeloid leukemia, associated with the Philadelphia chromosome translocation between chromosomes 9 and 22. In prenatal diagnostics, karyotyping is widely used for detecting chromosomal abnormalities in fetuses through procedures like amniocentesis and chorionic villus sampling. This allows for early identification of genetic disorders, enabling informed medical decisions and counseling for parents. In oncology, karyotyping helps in identifying chromosomal alterations in cancer cells, aiding in diagnosis, prognosis, and treatment planning. Despite its usefulness, karyotyping has limitations in detecting smaller genetic mutations, such as single-gene disorders or microdeletions, which require more advanced molecular techniques like fluorescence in situ hybridization (FISH) and chromosomal microarray analysis (CMA). However, karyotyping remains a crucial and widely used cytogenetic technique for diagnosing chromosomal abnormalities and understanding genetic disorders. You said. Genetic disorders, Downs ChatGPT said. Genetic disorders are conditions caused by abnormalities in an individual's DNA, which can result from mutations in a single gene, chromosomal alterations, or complex interactions between multiple genes and environmental factors. One of the most well-known genetic disorders is Down syndrome, also known as trisomy 21, which is caused by the presence of an extra copy of chromosome 21. This condition leads to characteristic physical features, intellectual disabilities, and an increased risk of various health complications. Down syndrome occurs due to an error in cell division known as non-disjunction, where chromosome 21 fails to separate properly during gamete formation. As a result, an individual inherits 3 copies of chromosome 21 instead of the usual 2. There are 3 main types of Down syndrome: trisomy 21, which accounts for about 95% of cases and results from a full extra chromosome 21 in every cell; mosaic Down syndrome, where only some cells have the extra chromosome, leading to milder symptoms; and translocation Down syndrome, where a piece of chromosome 21 attaches to another chromosome, usually chromosome 14, due to a structural rearrangement. The clinical features of Down syndrome include distinct facial characteristics such as a flat facial profile, upward slanting eyes, a small nose, and a protruding tongue. Affected individuals often have hypotonia, low muscle tone, short stature, and a single palmar crease in the hand. Cognitive impairments vary, with most individuals experiencing mild to moderate intellectual disability and delayed developmental milestones. Down syndrome is also associated with congenital heart defects, gastrointestinal abnormalities, hearing loss, vision problems, and an increased risk of thyroid dysfunction and leukemia. Prenatal screening and diagnostic tests are available to detect Down syndrome before birth. Screening tests, such as maternal serum markers and non-invasive prenatal testing (NIPT), assess the likelihood of the condition based on biochemical and DNA analysis of fetal cells in maternal blood. Confirmatory diagnostic tests, including chorionic villus sampling (CVS) and amniocentesis, provide definitive genetic information but carry a small risk of miscarriage. There is no cure for Down syndrome, but early intervention programs, medical management, and supportive therapies improve the quality of life for affected individuals. Specialized education, speech therapy, occupational therapy, and physical therapy help address developmental challenges, while regular medical checkups ensure early detection and treatment of associated health conditions. Advances in medical care have significantly increased the life expectancy of individuals with Down syndrome, with many living into their 60s. The clinical features of Down syndrome include distinct facial characteristics such as a flat facial profile, upward slanting eyes, a small nose, and a protruding tongue. Affected individuals often have hypotonia, low muscle tone, short stature, and a single palmar crease in the hand. Cognitive impairments vary, with most individuals experiencing mild to moderate intellectual disability and delayed developmental milestones. Down syndrome is also associated with congenital heart defects, gastrointestinal abnormalities, hearing loss, vision problems, and an increased risk of thyroid dysfunction and leukemia. Prenatal screening and diagnostic tests are available to detect Down syndrome before birth. Screening tests, such as maternal serum markers and non-invasive prenatal testing (NIPT), assess the likelihood of the condition based on biochemical and DNA analysis of fetal cells in maternal blood. Confirmatory diagnostic tests, including chorionic villus sampling (CVS) and amniocentesis, provide definitive genetic information but carry a small risk of miscarriage. There is no cure for Down syndrome, but early intervention programs, medical management, and supportive therapies improve the quality of life for affected individuals. Specialized education, speech therapy, occupational therapy, and physical therapy help address developmental challenges, while regular medical checkups ensure early detection and treatment of associated health conditions. Advances in medical care have significantly increased the life expectancy of individuals with Down syndrome, with many living into their 60s and leading fulfilling lives with appropriate support. Research on Down syndrome continues to focus on understanding its genetic basis, improving medical treatments, and enhancing cognitive and developmental outcomes. Studies exploring gene expression, potential drug therapies, and the role of neuroplasticity offer hope for future interventions that may mitigate the effects of the extra chromosome 21. You said, Down in detail. ChatGPT said. Down syndrome, a detailed overview. Introduction. Down syndrome, also known as trisomy 21, is a genetic disorder caused by the presence of an extra copy of chromosome 21. It is one of the most common chromosomal disorders, affecting approximately 1 in 700 live births worldwide. The condition is characterized by distinct physical features, developmental delays, intellectual disabilities, and an increased risk of certain medical conditions. While there is no cure, early intervention, medical care, and supportive therapy significantly improve the quality of life for individuals with Down syndrome. Causes and genetic basis. Down syndrome results from an error in chromosome segregation during cell division, leading to an extra chromosome 21. This abnormality occurs due to nondisjunction, translocation, or mosaicism. Trisomy 21, nondisjunction Down syndrome. This is the most common form, accounting for about 95% of cases. It occurs when chromosome 21 fails to separate properly during the formation of egg or sperm cells meiosis. As a result, the fertilized egg ends up with 3 copies of chromosome 21 instead of the usual 2. Translocation Down syndrome accounts for about 3–4% of cases. Occurs when an extra portion of chromosome 21 attaches to another chromosome, often chromosome 14, instead of being a separate extra chromosome. Unlike trisomy 21, this form can be inherited from a parent carrying a balanced translocation, meaning that they do not have Down syndrome but can pass a rearranged chromosome to their children. Mosaic Down syndrome. The rarest form, occurring in about 1–2% of cases. Happens when some cells in the body have the normal 46 chromosomes, while others have an extra chromosome 21. Since not all cells are affected, individuals with mosaic Down syndrome may have milder symptoms compared to those with full trisomy 21. Clinical features and symptoms. Individuals with Down syndrome have a combination of characteristic physical features, developmental delays, and medical conditions. 1. Physical features. Flat facial profile and small nose, upward slanting eyes with epicanthic folds, small ears and a short neck, protruding tongue due to a small oral cavity, hypotonia, low muscle tone leading to delayed motor development, short stature and shorter limbs. Single transverse palmar crease in the hand, small hands and feet with a wide gap between the first and second toes. 2. Intellectual and developmental characteristics. Intellectual disability ranging from mild to moderate, delayed speech and language development, slower cognitive processing and learning ability, short attention span and difficulty with problem solving, delayed motor skills such as sitting, standing, and walking. 3. Medical conditions associated with Down syndrome. Congenital heart defects, present in nearly 50% of affected individuals, including atrioventricular septal defect, AVSD, and ventricular septal defect, VSD. Gastrointestinal abnormalities, conditions like duodenal atresia, Hirschsprung disease, and gastroesophageal reflux are more common. Hearing and vision problems, higher risk of conductive hearing loss and vision disorders such as cataracts and strabismus. Thyroid dysfunction, hypothyroidism is frequently observed. Leukemia, increased risk of acute lymphoblastic leukemia, ALL, and acute myeloid leukemia, AML, in childhood. Obstructive sleep apnea, due to narrowed airways and muscle hypotonia. Alzheimer's disease, early onset dementia is more common due to the overexpression of amyloid precursor protein, AP, on chromosome 21. Diagnosis. Down syndrome can be diagnosed prenatally or after birth through a combination of screening and diagnostic tests. 1. Prenatal screening tests estimate the likelihood of Down syndrome but do not confirm it. First trimester screening measures levels of pregnancy-associated plasma protein A, PAP-A, and human chorionic gonadotropin, HCG, along with an ultrasound to check for nuchal translucency. Second trimester screening quad test measures levels of AFP, HCG, estriol, and inhibin A in maternal blood. Non-invasive prenatal testing, NIPT, analyzes fetal DNA in maternal blood for chromosomal abnormalities. 2. Diagnostic tests, definitive but invasive. Chorionic villus sampling, CVS, performed between 10 to 13 weeks of pregnancy, involving the extraction of placental tissue for chromosomal analysis. Amniocentesis, conducted between 15 to 20 weeks, where amniotic fluid is analyzed for fetal chromosomes. 3. Postnatal diagnosis. Physical examination, based on characteristic features. Karyotyping, confirms the presence of an extra chromosome 21. Management and treatment. Although there is no cure for Down syndrome, early intervention, medical care, and support can improve the quality of life for affected individuals. 1. Early intervention and education. Special education programs tailored to individual learning needs. Speech therapy, occupational therapy, and physical therapy to enhance communication and motor skills. 2. Medical management. Regular checkups with a pediatrician or genetic specialist. Cardiac evaluations for congenital heart defects. Routine thyroid function tests and hearing screenings. Vision assessments and corrective treatments for eye disorders. Nutritional support to maintain a healthy weight. 3. Social and emotional support. Support groups for families and caregivers. Encouragement of social inclusion and participation in community activities. Life skills training to promote independence. 4. Research and future treatments. Studies exploring gene therapy and potential drugs to enhance cognitive function. Advances in regenerative medicine and neuroplasticity research. Ongoing improvements in prenatal diagnostics for early detection. Prognosis and life expectancy. Due to advances in medical care, individuals with Down syndrome now have a significantly improved life expectancy, averaging around 60 years. With proper support, many individuals can lead fulfilling lives, participate in education and employment, and engage in social activities. While challenges exist, societal awareness, inclusivity, and medical advancements continue to enhance opportunities for people with Down syndrome, promoting their independence and well-being. You said, Klinefelter's ChatGPT said. Klinefelter syndrome, a detailed overview. Introduction. Klinefelter syndrome, KS, is a genetic disorder that affects males and is caused by the presence of an extra X chromosome, resulting in a karyotype of 47, XXY instead of the typical 46, XY. It is one of the most common chromosomal disorders in males, with an estimated occurrence of 1 in 500 to 1,000 live male births. The condition affects physical, cognitive, and reproductive development, leading to symptoms such as small testes, infertility, reduced testosterone levels, gynecomastia, breast enlargement, and learning difficulties. Despite these challenges, early diagnosis and management, including testosterone therapy and educational support, can significantly improve quality of life. Causes and genetic basis. Klinefelter syndrome occurs due to a chromosomal non-disjunction error during the formation of sperm or egg cells, leading to an extra X chromosome. This results in a 47, XXY karyotype instead of the normal 46, XY male chromosome complement. In some cases, additional X chromosomes e.g., 48, XXXY or 49, XXXXY can be present, leading to more severe symptoms. There are three main genetic variations of Klinefelter syndrome. Classic Klinefelter syndrome 47, XXY. The most common form, present in about 85–90% of cases. The extra X chromosome is present in all body cells. Mosaic Klinefelter syndrome 46, XY 47, XXY. Occurs when some cells have the extra X chromosome while others have the normal male karyotype. Symptoms tend to be milder, and fertility may be less severely affected. Variants with multiple X chromosomes 48, XXXY or 49, XXXXY. These rare forms lead to more pronounced intellectual disabilities and physical abnormalities. The extra X chromosome disrupts normal male development by interfering with testosterone production, leading to the characteristic features of the syndrome. Clinical features and symptoms. The symptoms of Klinefelter syndrome vary widely, and some individuals may have mild or no noticeable signs. The most prominent effects relate to reproductive health, physical development, and cognitive abilities. 1. Physical characteristics. Small, firm testes, testicular atrophy, leading to reduced testosterone production. Gynecomastia, breast enlargement, due to hormonal imbalance. Tall stature with long legs and a disproportionately shorter torso. Reduced facial and body hair compared to typical males. Decreased muscle mass and strength. Low bone density, increasing the risk of osteoporosis. Narrow shoulders and wider hips, giving a more feminized body shape. 2. Reproductive and endocrine features. Primary hypogonadism, failure of the testes to produce sufficient testosterone. Infertility due to low sperm production, azoospermia or oligospermia. Reduced libido and erectile dysfunction in some cases. Increased risk of testicular cancer. 3. Cognitive and behavioral characteristics. Mild to moderate learning disabilities, particularly in language and reading. Delayed speech and language development in childhood. Difficulty with executive functioning, such as organization and problem solving. Lower attention span and increased risk of attention deficit hyperactivity disorder, ADHD. Social difficulties, including shyness and social anxiety. 4. Associated health risks. Increased risk of metabolic disorders such as type 2 diabetes. Higher likelihood of developing autoimmune diseases like lupus and rheumatoid arthritis. Increased risk of cardiovascular disease and deep vein thrombosis. Thyroid dysfunction, particularly hypothyroidism. Greater susceptibility to breast cancer due to hormonal imbalances. Diagnosis. Klinefelter syndrome is often underdiagnosed due to its variable presentation. Many cases are not identified until adulthood, especially when individuals seek medical help for infertility. 1. Prenatal diagnosis. Can be detected through amniocentesis or chorionic villus sampling, CVS, during pregnancy if chromosomal analysis is performed. Non-invasive prenatal testing, NIPT, can also indicate the presence of an extra X chromosome. 2. Postnatal diagnosis. Physical examination. Doctors may suspect Klinefelter syndrome based on small testes, gynecomastia, and tall stature. Karyotyping, a blood test that confirms the presence of an extra X chromosome. Hormonal testing. Blood tests show low testosterone levels, elevated follicle-stimulating hormone, FSH, and luteinizing hormone, LH, indicating testicular dysfunction. Semen analysis reveals low or absent sperm count, confirming infertility in affected individuals. Management and treatment. There is no cure for Klinefelter syndrome, but various treatments and interventions can help manage symptoms and improve quality of life. 1. Hormone therapy. Testosterone replacement therapy (TRT) is the primary treatment, helping to develop male secondary sexual characteristics, increase muscle mass, improve mood and energy levels, and maintain bone density. TRT is usually started at puberty if the diagnosis is made early. 2. Fertility treatment. Most men with Klinefelter syndrome are infertile due to low sperm production. Assisted reproductive technologies are, like testicular sperm extraction (TEES) with intracytoplasmic sperm injection (ICSI), can help retrieve viable sperm for fertility treatment. 3. Speech and educational support. Early speech therapy helps with language delays. Special education programs assist with learning disabilities and academic challenges. Occupational therapy improves fine motor skills and coordination. 4. Psychological and social support. Counseling helps address emotional difficulties and self-esteem issues. Support groups provide assistance for affected individuals and their families. Social skills training can aid in overcoming communication challenges. 5. Management of associated health conditions. Regular checkups for osteoporosis, metabolic disorders, and cardiovascular health. Gynecomastia surgery may be considered for severe breast enlargement. Prognosis and life expectancy. With proper medical care and support, individuals with Klinefelter syndrome can lead healthy and fulfilling lives. Life expectancy is generally normal, but early detection and management of associated health risks improve overall well-being. Many affected individuals complete higher education, have successful careers, and maintain strong personal relationships. Advances in reproductive medicine also offer new possibilities for fertility preservation in some cases. Despite the challenges posed by Klinefelter syndrome, early diagnosis, medical intervention, and supportive therapies allow affected individuals to thrive and achieve a good quality of life. You said, Turner syndromes, ChatGPT said. Turner syndrome, a detailed overview introduction. Turner syndrome (TS) is a genetic disorder that affects females and is caused by the complete or partial absence of one X chromosome. It occurs in approximately 1 in 2,500 live female births. Turner syndrome leads to various developmental and medical challenges, including short stature, ovarian dysfunction, infertility, congenital heart defects, and certain learning difficulties. However, with early diagnosis and appropriate medical care, individuals with Turner syndrome can lead healthy and fulfilling lives. Causes and genetic basis. Turner syndrome results from a chromosomal abnormality in which one of the two X chromosomes is missing or altered in some or all cells. The condition arises due to errors in sperm or egg cell formation during meiosis, leading to one of the following karyotypic variations. Classic Turner syndrome 45X. The most common form, where one entire X chromosome is missing. Affects about 50–60% of individuals with Turner syndrome. Mosaic Turner syndrome 45, X46, XX or 45, X46, XY. Some have a normal 46, XX karyotype, while others have a missing X chromosome 45, X. Symptoms may be milder, depending on the proportion of affected cells. Partial X chromosome deletion. Instead of a complete absence, part of one X chromosome is missing or structurally altered. Severity of symptoms varies based on the extent of the deletion. Isochromosome and ring chromosome Turner syndrome. In some cases, one X chromosome is abnormally shaped, affecting gene expression. Unlike Klinefelter syndrome, which occurs only in males due to an extra X chromosome, Turner syndrome exclusively affects females and is one of the few viable monosomy conditions where only one sex chromosome is present instead of two. Clinical features and symptoms. Turner syndrome affects multiple systems in the body, leading to distinct physical, reproductive, and cognitive characteristics. 1. Physical characteristics. Short stature, average adult height without treatment, 4 feet 8 inches. Webbed neck, excess skin folds between the neck and shoulders. Low set ears and a small lower jaw. Broad chest with widely spaced nipples. Shorter fourth and fifth fingers. Lymphedema, swelling of hands and feet at birth. High arched palate and dental issues. 2. Reproductive and endocrine features. Gonadal dysgenesis, underdeveloped or non-functional ovaries, leading to little or no estrogen production. Delayed or absent puberty due to ovarian failure. Infertility, most individuals cannot conceive naturally. Lack of menstruation, primary amenorrhea. 3. Congenital heart defects and other medical. Most common form, where one entire X chromosome is missing. Affects about 50–60% of individuals with Turner syndrome. Mosaic Turner syndrome 45, X46, XX or 45, X46, XY. Some cells have a normal 46, XX karyotype, while others have a missing X chromosome 45, X. Symptoms may be milder, depending on the proportion of affected cells. Partial X chromosome deletion. Instead of a complete absence, part of one X chromosome is missing or structurally altered. Severity of symptoms varies based on the extent of the deletion. Isochromosome and ring chromosome Turner syndrome. In some cases, one X chromosome is abnormally shaped, affecting gene expression. Unlike Klinefelter syndrome, which occurs only in males due to an extra X chromosome, Turner syndrome exclusively affects females and is one of the few viable monosomy conditions where only one sex chromosome is present instead of two. Clinical features and symptoms. Turner syndrome affects multiple systems in the body, leading to distinct physical, reproductive, and cognitive characteristics. 1. Physical characteristics. Short stature, average adult height without treatment, 4 feet 8 inches. Webbed neck, excess skin folds between the neck and shoulders. Low set ears and a small lower jaw. Broad chest with widely spaced nipples. Shorter fourth and fifth fingers. Lymphedema, swelling of hands and feet at birth. High arched palate and dental issues. 2. Reproductive and endocrine features. Gonadal dysgenesis, underdeveloped or non-functional ovaries, leading to little or no estrogen production. Delayed or absent puberty due to ovarian failure. Infertility, most individuals cannot conceive naturally. Lack of menstruation, primary amenorrhea. 3. Congenital heart defects and other medical conditions. Bicuspid aortic valve, a common congenital heart defect leading to an increased risk of aortic stenosis. Coarctation of the aorta, narrowing of the aorta, requiring medical intervention. Hypertension, increased blood pressure is common. Kidney abnormalities, horseshoe kidney fusion of the kidneys is frequently observed. Osteoporosis, due to low estrogen levels, leading to brittle bones. Increased risk of type 2 diabetes and thyroid disorders. 4. Cognitive and behavioral characteristics. Normal intelligence but mild learning difficulties, especially in mathematics and spatial reasoning. Difficulty with visual spatial tasks, such as map reading and direction sense. Good verbal skills but challenges with nonverbal communication. Social difficulties, including shyness and difficulty interpreting social cues. Diagnosis. Turner syndrome can be diagnosed prenatally or after birth through various genetic tests. 1. Prenatal diagnosis. Non-invasive prenatal testing (NIPT) analyzes fetal DNA in maternal blood for chromosomal abnormalities. Amniocentesis, a definitive test that analyzes fetal chromosomes and amniotic fluid. Chorionic villus sampling (CVS) analyzes placental cells to detect missing or altered X chromosomes. 2. Postnatal diagnosis. Physical examination, based on characteristic physical features such as short stature and webbed neck. Karyotyping, confirms the presence of a missing or structurally altered X chromosome. Hormonal testing, reveals low estrogen levels and elevated follicle-stimulating hormone (FSH) and luteinizing hormone (LH) indicating ovarian failure. Echocardiogram and kidney ultrasound, used to detect heart and renal abnormalities. Management and treatment. There is no cure for Turner syndrome, but early medical intervention and supportive care help manage symptoms and improve quality of life. 1. Growth hormone therapy (GH). Administered in childhood to increase height and promote normal growth. Most effective when started early, ideally before puberty. 2. Estrogen replacement therapy (ERT). Begins at puberty to induce secondary sexual characteristics, such as breast development and menstruation. Essential for bone health and preventing osteoporosis. 3. Fertility treatment. Most individuals with Turner syndrome are infertile, but some may conceive using egg donation and assisted reproductive technologies (ART). In rare cases, mosaic Turner syndrome individuals may produce viable eggs. 4. Cardiovascular monitoring and treatment. Regular echocardiograms and blood pressure monitoring are crucial to detect and manage heart conditions. Surgical correction may be required for severe heart defects. 5. Management of associated conditions. Thyroid function tests to monitor for hypothyroidism. Calcium and vitamin D supplements to maintain bone health. Hearing tests to address possible hearing loss. 6. Psychological and educational support. Special education support for learning difficulties, particularly in math and spatial skills. Counseling and social skills training to enhance confidence in interpersonal relationships. Support groups for individuals and families affected by Turner syndrome. Prognosis and life expectancy. With proper medical care and hormonal treatment, individuals with Turner syndrome can lead normal, productive lives. Life expectancy is slightly reduced due to the increased risk of cardiovascular disease, but regular medical checkups and lifestyle modifications can improve long-term health. Many individuals with Turner syndrome complete higher education, pursue careers, and live fulfilling lives with strong social support. Although Turner syndrome presents various challenges, advancements in medical care, hormone therapy, and reproductive technologies have significantly improved outcomes for affected individuals. Early diagnosis and intervention play a crucial role in enhancing their quality of life. You said. Chromosomal aberrations and mutation. ChatGPT said. Chromosomal aberrations and mutation, a detailed overview. Introduction. Genetic variations arise due to changes in DNA sequence or chromosome structure. These changes, known as mutations and chromosomal aberrations, play a significant role in evolution, genetic diversity, and disease. While mutations involve alterations at the level of genes or nucleotides, chromosomal aberrations refer to large-scale structural or numerical changes in chromosomes. Both can have profound effects on an organism's phenotype and viability. 1. Chromosomal aberrations. Chromosomal aberrations involve changes in the structure or number of chromosomes, leading to genetic disorders and developmental abnormalities. These aberrations can be classified into two main types. Numerical aberrations, aneuploidy and polyploidy. Numerical chromosomal aberrations occur due to errors in chromosome segregation during cell division, mitosis or meiosis, resulting in an abnormal number of chromosomes. 1. Aneuploidy. Aneuploidy occurs when there is a gain or loss of individual chromosomes, leading to an abnormal chromosome number. This results from non-disjunction, where chromosomes fail to separate properly during meiosis. Monosomy 2n1, right pointing arrow 1 chromosome is missing, e.g., Turner syndrome, 45x. Trisomy 2n+1, right pointing arrow and extra chromosome is present, e.g., Down syndrome, 47, XX+21, Klinefelter syndrome, 47, XXY. Examples of aneuploidy in humans. Trisomy 21, Down syndrome, intellectual disability, heart defects, characteristic facial features. Trisomy 18, Edwards syndrome, severe developmental delay, congenital heart defects, short lifespan. Trisomy 13, Patau syndrome, cleft palate, polydactyly, severe brain and heart abnormalities. Turner syndrome, 45, X, short stature, ovarian failure, infertility. Klinefelter syndrome, 47, XXY, tall stature, gynecomastia, infertility. 2. Polyploidy. Polyploidy occurs when an organism has multiple sets of chromosomes, e.g., 3n, 4n. It is common in plants but rare in animals. Autopolyploidy, extra chromosome sets from the same species. Allopolyploidy, extra chromosome sets from hybridization between different species. B. Structural chromosomal aberrations. Structural abnormalities occur when chromosome segments are deleted, duplicated, inverted, or translocated due to breaks and rearrangements. 1. Deletion deficiency. A segment of a chromosome is lost, leading to loss of genetic material. Example, Cri-du-Chat syndrome, deletion in chromosome 5p causes intellectual disability and a characteristic high-pitched cry. 2. Duplication. A segment of a chromosome is repeated, increasing the dosage of certain genes. Example, Charcot-Marie Tooth disease, duplication of the PMP22 gene on chromosome 17 leads to progressive nerve degeneration. 3. Inversion. A chromosome segment is reversed end to end. Paracentric inversion does not involve the centromere. Paracentric inversion includes the centromere. Effects can cause infertility and abnormal chromosome pairing during meiosis. 4. Translocation. A chromosome fragment attaches to a non-homologous chromosome. Reciprocal translocation, exchange of segments between two chromosomes. Robertsonian translocation, fusion of two acrocentric chromosomes at the centromere. Example, chronic myeloid leukemia, CML, is caused by the Philadelphia chromosome, translocation between chromosomes 9 and 22, leading to BCR-ABL fusion. 2. Mutation. Mutations are changes in the DNA sequence that can be spontaneous or induced by environmental factors, mutagens. These alterations can affect a single nucleotide or large gene segments, leading to genetic disorders, cancers, and evolutionary adaptations. Types of mutations. 1. Point mutations, single nucleotide changes. Point mutations affect a single nucleotide in the DNA sequence. Substitutions, one base is replaced by another. Silent mutation, no change in the amino acid sequence, e.g., GAA right pointing arrow GAG, both coding for glutamate. Missense mutation, changes one amino acid in the protein, e.g., sickle cell anemia caused by blue right pointing arrow val substitution in hemoglobin. Nonsense mutation, creates a premature stop codon, leading to truncated proteins, e.g., Duchenne muscular dystrophy. 2. Frame shift mutations. Insertion or deletion of nucleotides shifts the reading frame, altering all downstream codons. Example, Tay-Sachs disease caused by frame shift mutation in the hexa gene, leading to fatal neurodegeneration. 3. Expanding repeats, trinucleotide repeat disorders. Short DNA sequences, e.g., CAG, CGG, are abnormally repeated, leading to unstable genes. Huntington's disease, expanded CAG repeats in the HTT gene cause progressive neurological degeneration. Fragile X syndrome, CGG repeat expansion in the FMR1 gene leads to intellectual disability. B. Causes of mutation. Mutations arise due to internal biological processes or external environmental factors. 1. Spontaneous mutations. Occur naturally due to errors in DNA replication, recombination, or repair. Example, DNA polymerase errors during cell division. 2. Induced mutations, mutagens. Mutagens are environmental agents that increase mutation rates. Chemical mutagens. Base analogs, e.g., 5-bromoaryl substitute for normal bases, causing indirect pairing. Alkylating agents, e.g., mustard gas modify bases, leading to mispairing. Intercalating agents, e.g., ethidium bromide insert into DNA, causing frameshift mutations. Physical mutagens. Ultraviolet UV radiation induces thymine dimers, leading to skin cancer. Ionizing radiation, X-rays, gamma rays causes DNA strand breaks and chromosomal mutations. Biological mutagens. Viruses, e.g., HPV, integrate into host DNA, causing mutations that lead to cancer. Conclusion. Chromosomal aberrations and mutations are key genetic changes that contribute to biodiversity, disease, and evolution. While some mutations provide adaptive advantages, others result in genetic disorders. Understanding these genetic alterations is crucial for diagnosing and managing hereditary diseases, developing therapies, and advancing biotechnology. With modern tools like gene therapy, CRISPR genome editing, and personalized medicine, scientists are now working to correct harmful mutations and prevent genetic disorders. You said. Deletion, duplication, inversion, translocation. ChatGPT said. Chromosomal aberrations, deletion, duplication, inversion, and translocation. Introduction. Chromosomal aberrations are structural changes in chromosomes that occur due to errors in DNA replication, repair, or recombination. These changes can significantly impact gene expression, leading to genetic disorders, cancer, or evolutionary variations. Structural aberrations include deletion, duplication, inversion, and translocation, all of which can disrupt normal genetic function. 1. Deletion. Deletion occurs when a segment of a chromosome is lost due to a break in the DNA. This results in the absence of certain genes, which can cause severe phenotypic effects if the missing genes are essential. Types of deletions. Terminal deletion. A single break occurs near the end of a chromosome, causing the loss of the distal fragment. Interstitial deletion. 2 breaks occur within a chromosome, and the intervening segment is lost. Effects of deletions. Loss of genetic material leads to gene dosage imbalance. Types of deletions. Terminal deletion. A single break occurs near the terminal deletion. Structural chromosomal aberrations, deletion, duplication, inversion, and translocation. Introduction. Chromosomal aberrations are structural changes in chromosomes that occur due to errors in DNA replication, repair, or recombination. These changes can significantly impact gene expression, leading to genetic disorders, cancer, or evolutionary variations. Structural aberrations include deletion, duplication, inversion, and translocation, all of which can disrupt normal genetic function. 1. Deletion. Deletion occurs when a segment of a chromosome is lost due to a break in the DNA. This results in the absence of certain genes, which can cause severe phenotypic effects if the missing genes are essential. Types of deletions. Terminal deletion. A single break occurs near the end of a chromosome, causing the loss of the distal fragment. Interstitial deletion. 2 breaks occur within a chromosome, and the intervening segment is lost. Effects of deletions. Loss of genetic material leads to gene dosage imbalance, affecting development and function. Haploinsufficiency. When a single copy of a gene is insufficient to produce a normal phenotype, leading to disease. Association with genetic disorders. Cree-Duchat syndrome. 5p deletion. Affected individuals have intellectual disability, microcephaly, and a characteristic cat-like cry. Wolff-Hirschhorn syndrome. 4p deletion causes developmental delays, seizures, and craniofacial abnormalities. Retinoblastoma. 13q deletion. Deletion of the RB1 tumor suppressor gene leads to eye cancer in children. 2. Duplication. Duplication occurs when a chromosome segment is copied and inserted into the genome, leading to extra genetic material. This results from unequal crossing over during meiosis or replication errors. Types of duplications. Tandem duplication. The duplicated segment is located adjacent to the original sequence. Reverse duplication. The duplicated segment is inverted in orientation. Displaced duplication. The duplicated segment is inserted at a different location in the same or another chromosome. Effects of duplications. Dosage effects. Increased expression of duplicated genes may alter cellular functions. Evolutionary significance. Gene duplications provide raw material for the evolution of new functions, e.g., hemoglobin gene family expansion. Associated disorders. Charcot-Marie Tooth disease. PMP22 gene duplication on chromosome 17 leads to peripheral nerve degeneration. MECP2 gene duplication syndrome causes severe intellectual disability in males. 3. Inversion. Inversion occurs when a chromosome segment is reversed end-to-end due to 2 breaks in the chromosome, followed by the reinsertion of the fragment in the opposite orientation. Inversions do not involve a loss or gain of genetic material but can disrupt gene function or cause reproductive issues. Types of inversions. Paracentric inversion. The inversion occurs outside the centromere, affecting only one chromosome arm. Paracentric inversion. The inversion includes the centromere, affecting both arms of the chromosome. Effects of inversions. Disruption of gene function. If an essential gene or regulatory region is inverted, gene expression may be altered. Reproductive consequences. In heterozygous individuals, crossing over within the inverted region during meiosis produces non-viable gametes due to chromosome misalignment. Associated conditions. Hemophilia A, caused by an inversion in the F8 gene on the X chromosome. Infertility. Some chromosomal inversions lead to reduced fertility due to abnormal chromosome segregation. 4. Translocation. Translocation occurs when a segment of one chromosome breaks and attaches to another non-homologous chromosome. It results from chromosome breakage and faulty repair mechanisms. Types of translocations. Reciprocal translocation, a mutual exchange of chromosome segments between two non-homologous chromosomes. Robertsonian translocation, the fusion of two acrocentric chromosomes with very short p arms, leading to a single large chromosome. This type of translocation commonly involves chromosomes 13, 14, 15, 21, and 22. Effects of translocations. Balanced translocations. If no genetic material is lost or gained, the individual may be phenotypically normal but may have reproductive issues. Unbalanced translocations. If there is a gain or loss of genetic material, the individual may have developmental disorders. Association with genetic disorders. Chronic myeloid leukemia, CML, caused by the Philadelphia chromosome, translocation between chromosomes 9 and 22, forming the BCR-ABL fusion gene. Burkitt's lymphoma, associated with a translocation between chromosomes 8 and 14, leading to overexpression of the MYC oncogene. Down syndrome translocation type, 46, XX or XY, T14, 21 caused by Robertsonian translocation involving chromosome 21. Conclusion. Structural chromosomal aberrations, deletion, duplication, inversion, and translocation, alter gene dosage, disrupt gene function, and contribute to various genetic disorders and evolutionary changes. While some chromosomal changes are lethal or disease-causing, others provide genetic diversity and evolutionary advantages. Advances in karyotyping, fluorescence in situ hybridization, FISH, and genomic sequencing have improved the diagnosis and understanding of these chromosomal abnormalities, enabling better genetic counseling and potential therapeutic interventions. You said. Ploidy and their genetic implications. ChatGPT said. Ploidy and its genetic implications. Introduction. Ploidy refers to the number of complete sets of chromosomes in a cell. It plays a fundamental role in genetics, evolution, and development. Organisms can be haploid, diploid, polyploid, or aneuploid, with each type affecting gene expression, reproduction, and phenotype. Changes in ploidy can have significant genetic consequences, including increased genetic variation, speciation, and disease. 1. Types of ploidy. Haploidy. N. Organisms are cells with only one complete set of chromosomes. Example, gametes, sperm and egg in sexually reproducing diploid organisms. Genetic implications. In haploid organisms, every mutation is immediately expressed since there is no second allele to mask its effect. Haploid stages allow natural selection to act directly on genetic variation. Many fungi and algae spend most of their life cycle in a haploid state. B. Diploidy. 2N. Organisms are cells with two sets of chromosomes, one from each parent. Example, most animals, including humans, are diploid 2n equals 46 in humans. Genetic implications. Diploidy allows genetic diversity through recombination during meiosis. It provides a backup copy of each gene, reducing the harmful effects of recessive mutations. Genetic diseases in diploids often follow Mendelian inheritance patterns. See polyploidy 3n, 4n, etc. Polyploidy occurs when an organism has more than 2 complete sets of chromosomes. It is common in plants but rare in animals. 1. Autopolyploidy. Arises from chromosome duplication within the same species, e.g., 3n, 4n, etc. Example, potatoes tetraploid, 4n equals 48. Genetic implications. Increased cell size and vigor, often resulting in larger fruits or flowers. Reduced fertility in odd-numbered polyploids, e.g., triploids like seedless bananas. Can lead to reproductive isolation and speciation. 2. Allopolyploidy. Results from hybridization between different species, followed by chromosome doubling. Example, wheat hexaploid, 6n equals 42, derived from hybridization events. Genetic implications. Allows the combination of beneficial traits from 2 species. Increases genetic diversity, leading to adaptability in new evolutionary lineages. Many allopolyploid plants are agriculturally important, e.g., wheat, cotton. D. Aneuploidy. Aneuploidy occurs when an organism gains or loses individual chromosomes, leading to an abnormal chromosome number. This results from non-disjunction during meiosis or mitosis. 1. Monosomy 2n1, loss of a single chromosome. Example, Turner syndrome 45, X in humans, where individuals have only one X chromosome. Genetic implications. Leads to severe developmental abnormalities or lethality. In Turner syndrome, individuals exhibit short stature, infertility, and cardiovascular defects. 2. Trisomy 2n plus 1. Addition of an extra chromosome. Examples. Down syndrome trisomy 21, intellectual disability, heart defects, and characteristic facial features. Klinefelter syndrome 47, XXY, males with an extra X chromosome, leading to infertility and gynecomastia. Genetic implications. Trisomies cause gene dosage imbalances, affecting development and metabolism. Most trisomies are lethal, except for a few, e.g., trisomy 21, 18, and 13. 2. Genetic implications of ploidy variations, a role in evolution. Polyploidy is a major force in plant evolution, leading to new species with increased genetic diversity. Many important crops, e.g., wheat, cotton, originated through polyploidization. Hybrid vigor in allopolyploids provides increased adaptability to different environments. B. Effects on gene expression. Polyploids often exhibit changes in gene regulation, leading to novel traits. Increased gene redundancy in polyploids can allow for evolutionary innovation and functional divergence. Aneuploidy often disrupts normal gene expression, leading to developmental defects or cancer. See impact on reproduction and fertility. Polyploids often face meiotic irregularities, making hybridization between diploids and polyploids difficult. Triploids 3n are usually sterile due to problems in chromosome pairing during meiosis, e.g., seedless watermelons. Aneuploidy can result in spontaneous abortions in humans, as most aneuploid embryos do not survive. D. Role in genetic disorders. Aneuploidy is linked to various human genetic disorders, e.g., Down syndrome, Turner syndrome, Klinefelter syndrome. Polyploidy is rare in humans but has been observed in certain cancers, where tumor cells exhibit abnormal chromosome numbers. In plants, polyploidy is often beneficial, leading to increased resilience and adaptation. Conclusion. Ploidy variations, ranging from haploidy to polyploidy and aneuploidy, have significant genetic consequences. While diploidy provides genetic stability, polyploidy drives evolutionary diversity, especially in plants. Aneuploidy, on the other hand, disrupts gene balance and leads to genetic disorders in animals. Understanding ploidy changes is crucial in genetics, evolutionary biology, agriculture, and medicine, offering insights into speciation, crop improvement, and genetic disease mechanisms. You said. Homologous and non-homologous recombination including transposition types. ChatGPT said. Homologous and non-homologous recombination, including transposition types. Introduction. Genetic recombination is a fundamental process in which DNA sequences are rearranged or exchanged, leading to genetic diversity, DNA repair, and genome evolution. There are 2 main types, homologous recombination and non-homologous recombination. Additionally, transposition represents a unique form of genetic recombination where mobile genetic elements move within the genome. 1. Homologous recombination, HR. Homologous recombination is the exchange of genetic material between 2 similar or identical DNA sequences. It is crucial for meiotic crossover, DNA repair, and maintaining genome stability. Mechanism of homologous recombination. Double-strand break formation. DNA is cleaved, often by ionizing radiation or endonucleases. Processing of DNA ends. Exonucleases degrade one strand, creating 3 feet single-stranded overhangs. Strand invasion. The single-stranded DNA invades a homologous duplex, forming a D-loop displacement loop. DNA synthesis and branch migration. DNA polymerase extends the invaded strand, and the holiday junction migrates. Resolution of holiday junctions. Cleavage at crossover points leads to either crossover or non-crossover products. Functions of homologous recombination. Genetic variation. During meiosis, HR allows recombination between maternal and paternal chromosomes. DNA repair. Repairs double-strand breaks via the homology-directed repair HDR pathway. Gene conversion. Unequal recombination can lead to non-reciprocal genetic exchange. Examples of homologous recombination. Meiotic recombination and gametogenesis. RECA-mediated recombination in bacteria, where RECA facilitates strand invasion. BRCA1, BRCA2 mediated repair in humans, preventing genomic instability and cancer. 2. Non-homologous recombination, NHR. Non-homologous recombination occurs without extensive sequence similarity between the recombining DNA molecules. It includes non-homologous end joining, NHEJ, and site-specific recombination. A non-homologous end joining, NHEJ repairs double-strand DNA breaks without a homologous template. Mechanism of NHEJ. Recognition of DNA breaks. The Cu70, Cu80 complex binds to broken DNA ends. End processing. DNA polymerases and nucleases modify ends for ligation. Ligation. DNA ligase IV seals the break, often leading to insertions or deletions, indels. Genetic implications of NHEJ. Fast but error-prone, causing mutations. Potential for VTJ recombination, generating diversity in antibodies and T-cell receptors. Frequently used by CRISPR-Cas9 gene editing, leading to targeted gene disruptions. B-site-specific recombination. Mediated by recombinases, e.g., Cre, FLP, integrases. Occurs at short, specific sequences, e.g., lambda phage integration into the E. coli genome. Used in gene therapy and genetic engineering, e.g., Krelok system for conditional gene knockouts. 3. Transposition, a special case of non-homologous recombination. Transposition is the movement of transposable elements, TEs, within the genome. TEs, or jumping genes, make up a significant portion of eukaryotic genomes. Types of transposition. A. Class 1. Retrotransposons copy and paste mechanism, move via an RNA intermediate. Reverse transcriptase converts RNA back into DNA, which integrates at a new site. Examples. Lines, long interspersed nuclear elements, autonomous, encode reverse transcriptase. Signs, short interspersed nuclear elements, non-autonomous, rely on lines for movement. Endogenous retroviruses, ERVs, related to retroviruses, persist in the genome. B. Class 2. DNA transposons cut and paste mechanism, move via DNA intermediate. Catalyzed by transposase, which cuts and reinserts the element elsewhere. Examples. P. Elements in Drosophila. A. C. D's elements in maize discovered by Barbara McClintock. C. Rolling circle transposons use a rolling circle replication mechanism. Examples. IS91 in bacteria. Genetic implications of transposition. Gene disruption. Insertions can inactivate essential genes, causing mutations. Genome evolution. TEs contribute to genetic diversity and regulatory changes. Human disease. Linked to cancers, line insertions in oncogenes, hemophilia, and Duchenne muscular dystrophy. Biotechnology. Used as tools for gene delivery, piggyback, sleeping beauty transposons. Conclusion. Homologous recombination ensures accurate genome maintenance and genetic diversity, whereas non-homologous recombination, including NHEJ and transposition, contributes to genome rearrangements, mutations, and evolution. Transposable elements play a significant role in shaping genomes, driving genetic innovation but also causing genomic instability. Understanding these recombination mechanisms is essential for genetics, medicine, and biotechnology applications. You said. Causes and detection of mutation, mutant types, lethal, conditional, biochemical, loss of function, ChatGPT said. Causes and detection of mutation, and types of mutants introduction. A mutation is any permanent alteration in the DNA sequence of an organism. Mutations can occur spontaneously due to errors in DNA replication or be induced by external factors such as radiation or chemicals. The effects of mutations range from silent changes with no impact to severe genetic disorders or lethality. Understanding their causes, detection methods, and types is essential for genetics, medicine, and evolutionary biology. 1. Causes of mutation. Mutations arise from spontaneous errors or environmental factors that damage DNA. Spontaneous mutations occur naturally due to cellular processes. Replication errors. DNA polymerase mistakes during replication lead to point mutations. Tautomeric shifts. Base modifications cause mispairing, e.g., thymine pairing with guanine. Strand slippage. Causes insertions or deletions common in repetitive sequences. B. Induced mutations. Caused by exposure to mutagens, chemical, physical, or biological agents that increase mutation rates. 1. Physical mutagens. Ionizing radiation, X-rays, gamma rays causes double-strand breaks, chromosomal aberrations. UV radiation induces pyrimidine dimers, TT bonds, leading to errors in DNA replication. 2. Chemical mutagens. Alkylating agents, e.g., EMS, mustard gas, add alkyl groups to bases, causing mispairing. Base analogs, e.g., 5-bromouracil incorporated into DNA instead of normal bases, leading to indirect base pairing. Interpolating agents, e.g., ethidium bromide, insert between DNA bases, causing frameshift mutations. 3. Biological mutagens. Transposons. Mobile genetic elements disrupt genes when they insert into DNA. Viruses. Some viruses, e.g., retroviruses, integrate into the genome, disrupting genes or causing cancers. 2. Detection of mutations. Several techniques help identify and study mutations in DNA. Molecular methods. PCR, polymerase chain reaction, amplifies DNA regions to detect small mutations. Sanger sequencing, determines the exact nucleotide sequence of DNA. Next-generation sequencing, NGS, identifies genome-wide mutations with high accuracy. Southern blotting, detects large insertions, deletions, and gene rearrangements. B. Cytogenetic methods. Karyotyping, identifies large chromosomal mutations, e.g., trisomies, translocations. Fluorescence in situ hybridization, FISH, detects specific gene deletions or amplifications. Comparative genomic hybridization, CGH, identifies copy number variations across the genome. C. Functional assays. Ames test, determines if a chemical induces mutations in bacteria. Gene expression analysis, RT-PCR, microarrays detects mutations affecting gene function. Protein studies, Western blot, mass spectrometry identifies mutations affecting protein structure. 3. Types of mutants. Mutants are classified based on their impact on survival, function, and environmental conditions. Lethal mutants. Mutations that cause death at some stage of development. Example, homozygous mutations in essential genes, such as mutations in the Tay-Sachs disease gene HEXA. B. Conditional mutants. Express phenotypic effects only under specific conditions. Example. Temperature-sensitive mutations. Certain mutations allow protein function at normal temperatures but inactivated at higher temperatures, e.g., TS mutations in viruses like influenza. C. Biochemical mutants, auxotrophs. Unable to synthesize an essential molecule and require it in the medium. Example. Histidine auxotrophs in bacteria need histidine to grow because of mutations in biosynthetic genes. D. Loss of function, LOF mutants. Function, LOF mutants. Mutations that reduce or eliminate gene activity. Example. CFTR mutations in cystic fibrosis lead to defective chloride ion transport. E. Gain of function, GOF mutants. Mutations that increase or alter protein function. Example. Mutant P53 oncogene leads to uncontrolled cell division in cancers. Conclusion. Mutations occur due to spontaneous errors or environmental mutagens, leading to genetic variation or diseases. They are detected using molecular, cytogenetic, and functional methods. Mutants can be lethal, conditional, biochemical, loss of function, or gain of function, each with distinct biological effects. Understanding these mutations is crucial for genetics, medicine, and biotechnology. You said. Gain of function, germinal versus somatic mutants, insertional mutagenesis. ChatGPT said. Gain of function mutations, germinal versus somatic mutants, and insertional mutagenesis introduction. Mutations can have different effects on gene function and inheritance patterns. Gain of function mutations enhance or create a new function for a gene, whereas loss of function mutations lead to reduced or absent gene activity. Germinal and somatic mutations differ in their transmission to offspring. Insertional mutagenesis occurs when foreign DNA integrates into a genome, disrupting normal gene function. Understanding these concepts is crucial in genetics, disease mechanisms, and biotechnology. 1. Gain-of-function (GOF) mutations. A gain-of-function mutation results in a gene acquiring a new function, increased activity, or expression in an abnormal context. These mutations often lead to dominant phenotypes, since a single mutated allele can alter cellular processes. Characteristics of gain-of-function mutations increase gene activity, overexpression or enhanced protein function. New protein function. A mutated protein may gain a novel capability. Altered gene regulation. The gene may be expressed at inappropriate times or in the wrong tissues. Often dominant. One mutated copy can be sufficient to cause an effect. Examples of gain-of-function mutations: cancer-causing oncogenes. Mutant ROS proteins remain constantly active, leading to uncontrolled cell division. Mutant p53 proteins acquire new functions, promoting tumor progression. Achondroplasia, dwarfism. FGFR3 mutations lead to excessive inhibition of bone growth. Huntington's disease. Expanded CAG repeats in the HTT gene cause toxic protein accumulation. 2. Germinal versus somatic mutants. Mutations can occur in germ cells, sperm and eggs, or somatic body cells, leading to different consequences for inheritance and disease. A germinal germline mutants occur in gametes, sperm or egg cells, and are heritable. Can be passed to all cells of the offspring, affecting the next generation. Responsible for inherited genetic disorders. Examples. Sickle cell anemia. A germline mutation in the HBB gene is passed from parents to offspring. Huntington's disease. A trinucleotide repeat expansion in HTT, inherited from an affected parent. BRCA1, BRCA2 mutations, increase the risk of breast and ovarian cancer. B. Somatic mutants. Occur in body cells after fertilization and are not inherited. Confined to the individual and may lead to cancer, mosaicism, or localized genetic disorders. The effects depend on when and where the mutation occurs. Examples. Skin cancer, melanoma, due to UV-induced mutations in TP53. Colorectal cancer caused by mutations in the APC gene in somatic cells. Segmental mosaicism, patchy genetic variation in an individual. Comparison of germline versus somatic mutations. Germline mutations, right pointing arrow, heritable, affects all cells in offspring. Somatic mutations, right pointing arrow, non-heritable, affects only specific tissues in an individual. 3. Insertional mutagenesis. Insertional mutagenesis occurs when foreign DNA, such as a transposable element, viral genome, or artificially introduced DNA, integrates into a host genome, disrupting gene function. Causes of insertional mutagenesis: transposons, jumping genes. Move within the genome, inserting into genes and disrupting their function. Example, the P element in Drosophila causes mutations by disrupting genes. Retroviral integration. Some viruses, like HIV or HTLV, integrate their DNA into host chromosomes, altering gene expression. Example, HTLV1 integration into T cells can lead to leukemia. Gene therapy vectors. Viral vectors used in gene therapy can integrate into oncogenes or tumor suppressor genes, leading to unintended mutations. Example, some patients in early gene therapy trials for SCID, severe combined immunodeficiency, develop leukemia due to vector insertion near oncogenes. Consequences of insertional mutagenesis. Loss of function mutations. If the insertion disrupts an essential gene, it can cause disease. Gain of function effects. Insertion near regulatory regions can increase gene expression abnormally. Chromosomal instability. Random insertions can lead to deletions, duplications, or translocations. Conclusion. Gain of function mutations enhance or create new gene activities, often leading to dominant phenotypes, as seen in cancers and certain genetic disorders. Germline mutations are heritable and affect offspring, while somatic mutations impact only the individual. Insertional mutagenesis results from the integration of foreign DNA into the genome, causing disruptions in gene function and leading to diseases such as cancer or genetic disorders. Understanding these mutations is essential for genetics, biotechnology, and medical research. You said. Transposable genetic elements, transposition and transposons, ChatGPT said. Transposable genetic elements, transposition and transposons. Introduction. Transposable genetic elements, also known as transposons or jumping genes, are DNA sequences capable of moving from one genomic location to another. They were first discovered by Barbara McClintock and Mays and are now recognized as major contributors to genetic variation, genome evolution, and mutations. Transposition refers to the mechanism by which transposons move within the genome, sometimes disrupting genes or altering their expression. 1. Transposable elements and their types. Transposable elements are classified into two major types based on their mode of movement. A Class 1, retrotransposons copy and paste mechanism, move via an RNA intermediate using a reverse transcriptase enzyme. The transposon is first transcribed into RNA, which is then reverse transcribed into DNA and inserted into a new genomic location. Since the original copy remains in place, retrotransposons tend to increase genome size over time. Types of retrotransposons. Long-terminal repeat LTR. Retrotransposons contain LTRs at both ends, similar to retroviruses. Example, human endogenous retroviruses, HERVs. Non-LTR retrotransposons. Lack LTRs and move using an endonuclease-mediated mechanism. Example. Lines long interspersed nuclear elements encode reverse transcriptase, can move autonomously. Signs short interspersed nuclear elements do not encode reverse transcriptase, rely on lines for mobility, e.g., ALU elements in humans. B Class 2, DNA transposons, cut and paste mechanism. Move directly as DNA without an RNA intermediate. The transposase enzyme cuts the transposon from one location and integrates it into another. This process does not increase genome size but can cause genetic instability. Types of DNA transposons. Autonomous transposons. Encode their own transposase enzyme, allowing independent movement. Example, P elements in Drosophila. Non-autonomous transposons. Lack transposase genes and depend on autonomous transposons for movement. Example, ACTs elements in Mays, activators and dissociators. 2. Mechanisms of transposition. Transposition can occur through different mechanisms, depending on whether the transposon moves via DNA or RNA. A retrotransposition, RNA-mediated, Class 1 transposons, transposon is transcribed into RNA. Reverse transcriptase converts RNA into complementary DNA, cDNA. cDNA is inserted into a new genomic location. This leads to copy number expansion of the transposon, common in eukaryotic genomes, e.g., ALU and line elements in humans. BDNA transposition, cut and paste, Class 2 transposons, transposase enzyme recognizes terminal sequences and excises the transposon. The transposon is moved to a new site and inserted into the genome. The DNA strand is repaired at the original site, sometimes leaving a footprint mutation. Unlike retrotransposons, DNA transposons do not replicate themselves. Common in bacteria and some eukaryotes, e.g., is elements in prokaryotes. 3. Effects and significance of transposons. Transposable elements play important roles in genome evolution, genetic diversity, and disease. A genomic instability and mutations. Insertion of transposons into coding or regulatory regions can disrupt gene function, leading to mutations. Example, hemophilia A caused by an ALU insertion in the factor VIII gene. B. Gene regulation and evolution. Transposons can modify gene expression by inserting near promoters or enhancers. Some transposons have been co-opted into regulatory elements, influencing development in immune system genes. See human diseases linked to transposons. Line 1 insertions are associated with certain cancers. ALU repeats contribute to genetic disorders by causing unequal crossover events. D. Biotechnology and gene manipulation. Transposons are used in genetic engineering, such as the Sleeping Beauty transposon system for gene therapy. Mutagenesis studies use transposons to disrupt genes and study their functions in model organisms. Conclusion. Transposable genetic elements are dynamic components of the genome that can move via retrotransposition, RNA-based, class 1 or DNA transposition, cut and paste, class 2 mechanisms. While they can cause mutations and genome instability, they also play essential roles in genome evolution, gene regulation, and biotechnology. Understanding transposons provides insights into genetic diversity, human diseases, and potential therapeutic applications. You said. Class 1 and Class 2. Transposable elements, LTR and non-LTR retrotransposons, ChatGPT said. Class 1 and Class 2 transposable elements, LTR and non-LTR retrotransposons. Introduction. Transposable elements, TEs, or jumping genes, are mobile genetic elements that can move within a genome. They are classified into two main types based on their mode of transposition, Class 1 retrotransposons and Class 2 DNA transposons. Retrotransposons use an RNA-mediated, copy and paste, mechanism, while DNA transposons use a cut and paste mechanism. 1. Class 1 transposable elements, retrotransposons, RNA-mediated. Retrotransposons move through an RNA intermediate and require reverse transcriptase to convert RNA back into DNA before reinsertion into the genome. They are the most abundant transposable elements in eukaryotic genomes and increase genome size due to the replication-based transposition. Types of retrotransposons. Class 1 elements are divided into LTR and non-LTR retrotransposons, based on the presence or absence of long terminal repeats, LTRs. Class 1 elements are divided into LTR and non-LTR retrotransposons, based on the presence or absence of long terminal repeats, LTRs. LTR retrotransposons contain long terminal repeats, LTRs, at both ends. Encode reverse transcriptase and integrase, similar to retroviruses. Move by being transcribed into RNA, reverse transcribed into DNA, and inserted back into the genome. Found in plants, fungi, and animals. Examples of LTR retrotransposons: endogenous retroviruses, ERVs. Ancient viral remnants in the genome, contributing to gene regulation. Example, HERV, human endogenous retrovirus, in the human genome. Tie elements in yeast. Well-studied LTR transposons in Saccharomyces cerevisiae. Copia and Gypsy elements in Drosophila, two major classes of LTR transposons in fruit flies. B. Non-LTR retrotransposons lack long terminal repeats, LTRs. Move through target prime reverse transcription, TPRT, often autonomous or non-autonomous. Types of non-LTR retrotransposons: lines, long interspersed nuclear elements, autonomous, encode reverse transcriptase. Example, line 1, L1, in humans, associated with some cancers. Signs, short interspersed nuclear elements, non-autonomous, rely on lines for transposition. Example, ALU elements in humans, the most abundant transposable elements. 2. Class 2 transposable elements, DNA transposons, DNA-mediated. DNA transposons move directly as DNA through a cut-and-paste mechanism without an RNA intermediate. They encode a transposase enzyme, which recognizes specific sequences, excises the transposon, and integrates it into a new location. Characteristics of DNA transposons: move via a direct cut-and-paste mechanism. Do not replicate themselves during transposition, unlike retrotransposons. Less common in humans but found in bacteria, plants, and some animals. Can cause mutations if they insert into functional genes. Types of DNA transposons. Autonomous DNA transposons encode their own transposase enzyme, allowing independent movement. Example, P elements in Drosophila. Non-autonomous DNA transposons lack transposase and require autonomous elements for movement. Example, ACD system in maize activator dissociation elements. Examples of DNA transposons. Is elements insertion sequences in bacteria, simple DNA transposons carrying only the transposase gene. TN transposons in bacteria. Larger DNA transposons carrying additional genes, e.g., antibiotic resistance. Sleeping Beauty transposon, artificially reactivated, used in genetic engineering and gene therapy research. 3. Comparison of Class 1 and Class 2 transposable elements. Feature: Class 1 retrotransposons, Class 2 DNA transposons, mode of transposition RNA intermediate, copy and paste, direct DNA movement, cut and paste, key enzyme, reverse transcriptase, transposase. Effect on genome size: increases genome size, multiple. Class 1 retrotransposons, LTR and non-LTR, replicate via an RNA intermediate, expanding the genome, while Class 2 DNA transposons move through a cut and paste mechanism without increasing genome size. These elements influence gene expression, cause mutations, and have applications in biotechnology and genetic research. You said. P elements in Drosophila, is. Elements in bacteria, composite transposons, TN family, medical significance. ChatGPT said. P elements in Drosophila, is elements in bacteria, composite transposons, TN family, and medical significance. Introduction. Transposable elements, TEs, are mobile genetic elements that move within genomes. They are classified into Class 1 retrotransposons and Class 2 DNA transposons. Among Class 2 elements, several important transposons include P elements in Drosophila, is elements in bacteria, composite transposons, and the TN family. These elements play significant roles in genome evolution, mutagenesis, antibiotic resistance, and medical research. 1. P elements in Drosophila discovery and characteristics. P elements are class II transposable elements found in the fruit flight Drosophila melanogaster. Discovered in the 1970s, they were recognized as the cause of hybrid dysgenesis, a phenomenon in which crosses between certain strains of Drosophila lead to genetic instability. They move via a cut-and-paste mechanism using the P-transposase enzyme. Significance in genetics. Mutagenesis tool. P elements are used for insertional mutagenesis in genetic studies. Transgenic applications. They serve as vectors for gene transfer and genetic engineering in Drosophila. P elements are class II transposable elements found in the fruit flight Drosophila melanogaster. Discovered in the 1970s, they were recognized as the cause of hybrid dysgenesis, a phenomenon in which crosses between certain strains of Drosophila lead to genetic instability. They move via a cut-and-paste mechanism using the P-transposase enzyme. Significance in genetics. Mutagenesis tool. P elements are used for insertional mutagenesis in genetic studies. Transgenic applications. They serve as vectors for gene transfer and genetic engineering in Drosophila. Hybrid dysgenesis. When P element carrying males mate with P element free females, uncontrolled transposition occurs, leading to sterility and chromosomal damage. Applications in research. P elements help study gene function and regulation in model organisms. Used in P element mediated transformation, a technique for introducing foreign genes into Drosophila. 2. Is elements in bacteria. Insertion sequences is the simplest transposons as elements are the simplest type of transposable elements in prokaryotes. They contain only the genes necessary for transposition, primarily encoding transposase. Typically 700 to 500 base pairs long and flanked by inverted repeat sequences. Mechanism of action. Move via cut-and-paste mechanism, excising themselves and reinserting elsewhere. Can disrupt genes and alter gene expression if inserted into functional regions. Examples. Is1, Is2, Is3, and Is10 are common as elements found in Escherichia coli and other bacteria. Is50 is part of TN5, a composite transposon carrying an antibiotic resistance gene. Significance in bacterial evolution. Cause gene disruptions and mutations. Influence bacterial genome rearrangements. Provide sites for homologous recombination, increasing genetic variability. 3. Composite transposons. Definition and structure. Composite transposons consist of two as elements flanking a central gene of interest, usually an antibiotic resistance gene. The as elements provide the transposase enzyme, allowing the entire transposon to move. Examples. TN5 contains kanamycin resistance flanked by as50 elements. TN9 carries chloramphenicol resistance, flanked by as1 elements. TN10 provides tetracycline resistance, flanked by as10 elements. Importance. Enable bacteria to acquire antibiotic resistance by horizontal gene transfer. Cause genetic rearrangements in bacterial genomes. Play a role in adaptive evolution and pathogenicity. 4. TN family of transposons definition and characteristics. TN transposons are larger complex transposons that carry genes beyond those required for transposition, often antibiotic resistance genes. Unlike as elements, they encode additional proteins involved in drug resistance and survival. Examples of TN transposons. TN3 carries beta-lactamase, conferring ampicillin resistance. TN5 provides kanamycin and streptomycin resistance. TN7 known for its site-specific insertion and antibiotic resistance properties. TN10 confers tetracycline resistance. Role in bacterial evolution. Contribute to the spread of multi-drug resistance (MDR). Involved in horizontal gene transfer through conjugation and transformation. Promote genomic plasticity in bacteria, aiding in adaptation to antibiotics. 5. Medical significance of transposable elements. A role in antibiotic resistance. TN transposons and composite transposons contribute to antibiotic resistance gene transfer in bacteria. Example, TN3 transposon in E. coli confers ampicillin resistance, making infections harder to treat. Multidrug-resistant pathogenic bacteria, e.g., MRSA, MDR-TB, acquire resistance genes through transposons. B transposons in genetic diseases in humans. Line 1 and ALU elements cause spontaneous mutations in humans. Insertional mutagenesis by transposons can lead to cancers, e.g., colon cancer, breast cancer, and neurological disorders. C transposons in gene therapy and biotechnology. Sleeping Beauty transposon system is used for gene delivery and gene therapy. Piggyback transposon is applied in stem cell research and genome editing. Transposons are used to create mutant libraries for genetic studies. Conclusion. Transposable elements such as P elements in Drosophila, Is elements in bacteria, composite transposons, and the TN family play crucial roles in genome evolution, genetic instability, antibiotic resistance, and disease development. While they pose challenges in bacterial infections and human genetic diseases, they are also powerful tools in biotechnology, genetic research, and gene therapy. Understanding their mechanisms and impact is essential for combating drug resistance and advancing medical science. Search. Rhizo. The immune system is a complex network of cells, tissues, and organs that work together to defend the body against infections, toxins, and abnormal cells. The fundamental components of this system include various immune cells, which originate from hematopoietic stem cells, HSCs, and specialized organs that support immune cell development and function. Hematopoiesis, origin of immune cells. Hematopoiesis is the process by which all blood cells, including immune cells, are formed. It occurs primarily in the bone marrow, where multipotent hematopoietic stem cells, HSCs, differentiate into two major progenitor lineages. Lymphoid lineage, adaptive and some innate immunity. B cells, responsible for antibody production, humoral immunity. T cells, mediators of cell-mediated immunity. Natural killer NK cells, cytotoxic lymphocytes involved in innate immunity. Myeloid lineage, innate immunity and precursor to red blood cells and platelets. Granulocytes. Neutrophils, first responders to infection, phagocytic. Eosinophils, combat parasitic infections, involved in allergic reactions. Basophils, release histamine, involved in allergic responses. Monocytes, macrophages. Monocytes circulate in the blood and differentiate into macrophages in tissues, acting as professional phagocytes. Dendritic cells, DCs, antigen-presenting cells, APCs, that initiate adaptive immune responses. Mast cells, release histamine and other mediators in allergic reactions. Megakaryocytes, give rise to platelets involved in clotting. Erythrocytes, red blood cells, RBCs, primarily oxygen transport but also play roles in immune signaling. Primary and secondary immune organs. Immune organs are categorized into primary, sites of immune cell development, and secondary, sites of immune activation and response. Primary, central, immune organs, bone marrow. Site of hematopoiesis, generation of all blood cells. B-cell maturation occurs here. Thymus. Site of T-cell maturation and selection. Ensures T-cells develop self-tolerance, prevention of autoimmunity. Secondary, peripheral, immune organs, lymph nodes. Filter lymphatic fluid for antigens. House B and T-cells, site of adaptive immune response initiation. Spleen. Filters blood for pathogens and damaged cells. Contains red pulp, RBC clearance, and white pulp, lymphocyte-rich areas. Mucosa-associated lymphoid tissues, MALT. Includes Peyer's patches, gut-associated tonsils, and bronchial-associated lymphoid tissues. Defends mucosal surfaces against pathogens. Other lymphoid tissues. Gut-associated lymphoid tissue, bulk, includes Peyer's patches, appendix, and lymphoid aggregates in the intestines. Bronchus-associated lymphoid tissue, bulk, defends respiratory tract. Functional roles of immune cells. Neutrophils and macrophages, phagocytose pathogens and debris. Dendritic cells, present antigens to naive T cells, initiating adaptive immunity. B cells, differentiate into plasma cells that secrete antibodies. T cells, include cytotoxic T cells, kill infected cells, and helper T cells coordinate immune responses. NK cells, recognize and destroy virally infected and cancerous cells. Practice questions. Multiple choice questions, MCQs. Which of the following immune cells originates from the myeloid lineage? A. B cells. B. T cells. C. Natural killer NK cells. D. Neutrophils. Answer. D. Neutrophils. Which organ is responsible for the maturation of T lymphocytes? A. Bone marrow. B. Spleen. C. Thymus. D. Lymph nodes. Answer. C. Thymus. Which secondary lymphoid organ is primarily responsible for filtering blood? A. Lymph nodes. B. Spleen. C. Bone marrow. D. Peyer's patches. Answer. B. Spleen. Previous year question. Which of the following is not a secondary lymphoid organ? A. Spleen. B. Lymph node. C. Thymus. D. Peyer's patches. Answer. C. Thymus. The thymus is a primary lymphoid organ, where T-cell maturation occurs, while the others are secondary lymphoid organs involved in immune responses. This completes the detailed coverage of cells and organs of the immune system, focusing on hematopoiesis, immune cell lineages, and lymphoid organs essential for immune function. Let me know if you need modifications or the next topic. You said. Regulation of hematopoiesis. ChatGPT said. Regulation of hematopoiesis. Hematopoiesis is a tightly regulated process that ensures the continuous production of blood cells from hematopoietic stem cells, HSCs, in the bone marrow. The regulation of hematopoiesis is crucial for maintaining immune homeostasis, responding to infections, and replenishing blood cell populations. This regulation is controlled by a combination of intrinsic factors, genetic and epigenetic control, extrinsic factors, cytokines and growth factors, and microenvironmental signals, bone marrow niche interactions. 1. Hematopoietic stem cells, HSCs, and their regulation. Hematopoietic stem cells, HSCs, are multipotent cells capable of self-renewal and differentiation into all blood cell lineages. Their regulation occurs through several key mechanisms. A. Transcriptional regulation. Transcription factors play a vital role in determining cell fate by regulating gene expression. Some key transcription factors include RUNX1 essential for HSC formation and differentiation. GATA1 drives erythroid and megakaryocyte differentiation. 2.1 regulates myeloid, monocyte, macrophage, dendritic cell, and lymphoid differentiation. Ikaros and Notch signaling, critical for lymphoid lineage commitment. CEBP, CCAAT, enhancer binding proteins involved in myeloid lineage differentiation. These transcription factors work in a network, ensuring the correct lineage commitment and functional maturation of immune cells. B. Epigenetic regulation. Epigenetic mechanisms such as DNA methylation, histone modifications, and non-coding RNAs control gene accessibility and expression. DNA methylation by DNMTs, DNA methyltransferases, regulates lineage-specific gene activation or suppression. Histone modifications like acylation and methylation determine chromatin accessibility for transcription factors. MicroRNAs, mRNAs, fine-tune gene expression by targeting mRNA for degradation or translational repression. These epigenetic modifications ensure hematopoiesis is dynamic and adaptable to physiological needs. 2. Extrinsic regulation, cytokines and growth factors. Extrinsic signals, primarily cytokines and growth factors, play an essential role in directing HSC self-renewal and differentiation. The major regulators include stem cell factor, SCF, c-KIT ligand supports HSC survival and self-renewal. Interleukins, ILs. IL-3 stimulates myeloid progenitors. IL-7 essential for lymphoid lineage commitment, B and T cells. IL-5 promotes eosinophil differentiation. Granulocyte macrophage colony stimulating factor, GM-CSF, stimulates granulocyte and monocyte differentiation. Granulocyte colony stimulating factor, GCSF, drives neutrophil production. Macrophage colony stimulating factor, MCSF, promotes monocyte macrophage differentiation. Erythropoietin, EPO, produced by the kidneys, stimulates erythroid progenitor expansion. Thrombopoietin, TPO, essential for megakaryocyte platelet precursor development. These cytokines act via receptor-mediated signaling pathways, often involving JAK-STAT, Janus kinase signal transducer and activator of transcription pathways, MAPK, mitogen-activated protein kinase, and PI3K ACT signaling, to drive lineage-specific differentiation. 3. Bone marrow microenvironment, niche regulation. The bone marrow provides a specialized microenvironment, or niche, that supports HSC maintenance and differentiation. It consists of osteoblastic endosteal niche, located near the bone surface, maintains HSC quiescence dormancy. Vascular niche, associated with blood vessels, supports HSC activation and mobilization. Key components of the niche include mesenchymal stromal cells, MSCs, provide support via cytokines like SC.